This is referred to as the missense mutation. Because the chemical sense sequence of amino acids and the ensuing polypeptide has changed, if the change occurred in a critical point in the polypeptide chain, the protein could be inactive or have reduced activity. However, not all mutations have caused amino acid substitution necessary to lead to non-functional proteins. The outcome depends on where in the polypeptide chain the substitution has occurred and on how it affects the folding and the catalytic activity of the protein. A missense mutation can lead to an enzyme that is temperature sensitive. Temperature sensitive mutants of bacteria are known, for example, that function normally at 30 degrees Celsius but cannot grow at 40 degrees Celsius. Although the wild type grows well at both temperatures, such mutations are also called conditionally lethal. Because the bacteria cannot grow under one condition but can under another, temperature-sensitive phenotypes often occur because the mutant protein can maintain its correct conformation at the low temperature but becomes partially unfolded, denatured at a high temperature. Another possible result of the base pair substitution is the formation of stop codon, which results in a premature termination of translation, leading to an incomplete protein that would almost certainly not be functional. Mutations of this type are called nonsense mutations because the change is from a codon for an amino acid, sense codon, to a stop codon, nonsense codon. Unless the nonsense mutations occurs very near the end of the gene, the incomplete protein would be completely inactive. Frameshift mutations. Because the genetic code is read from one end in consecutive blocks of 3 bases, and deletion or insertion of a base pair results in the reading frame shift, then the translation of the gene is completely upset. Partial restoration of gene function can often be accomplished by insertion of another base pair. Near the one deleted, one kind of suppressor mutation. See later in this section, after correction depending on the exact amino acids coded by the still faulty region and the region of the protein involved. And the protein formed may have some biological activity or even be completely normal. It is important to remember that micro-insertions or micro-deletions are frameshift mutations only if they occur in the part of protein encoding gene that encodes the reading frame. A single base pair insertion in the promoter or of a gene could lead to a dramatic change in the ability of the gene to function because of increased or decreased expression or a change in regulation. But it would not be a frameshift mutation. Similarly, base pair substitutions that are not within the reading frame are not missense or nonsense mutations. Back mutations or reversions. Point mutations are reversible. A revertant is operationally defined as a strain in which the wild-type phenotype that was lost in the mutant is restored. Revertants can be of 2 types. In same-site revertants, the mutation that restores activity occurs at the same site at which the original mutation occurred. If the back mutation is not only at the same site, but also leads to the wild-type sequence, it is called the true revertant. In second-site revertants, the mutation occurs at a different site in the DNA. Second-site mutations may cause restoration of a wild-type phenotype because of several types of suppressor mutations that restore the original phenotype. Suppressor mutations are new mutations that compensate for the effect of the original mutation. Several types of suppressor mutations are known. 1. A mutation somewhere else in the same gene can restore enzyme function, such as the frameshift mutation. 2. A mutation in another gene may restore the function of the original mutated gene and the wild type phenotype. And 3. A mutation, another gene that may result in the production of another enzyme that can replace the mutant one by introducing a metabolic pathway different from that used by the mutant enzyme. In this last type, no production of the original enzyme occurs, although it does in other types. Mutations involving many base pairs. Deletions are mutations in which a region of DNA has been eliminated. As we have discussed in microdeletions, the removal of one or very few base pairs are often frameshift mutations and can inactivate a gene. However, deletions can also involve the loss of hundreds and thousands of base pairs. Deletion of a large segment of a DNA results in a complete loss of function of any gene that may be involved. Some deletions are so large that they involve several genes. If any of the genes are essential, the mutations will be lethal. Such deletions cannot be restored through the further mutations, but only through genetic recombination. Indeed, one way in which large deletions are distinguished from point mutation is that the latter are reversible through further mutations, whereas the former are not. Insertions occur when new bases are added to the DNA. As we have discussed, insertions like deletions can involve only a single base or many bases. Microinsertions result from replication errors as do deletions, but larger insertions arise as a result of mistakes that occur during genetic recombination. Insertions inactivate the gene in which they occur. Many insertion mutations are due to the insertion of specific identifiable DNA sequences, 700 to 1400 base pairs in length, called insertion sequences, a type of transposable element. The behavior of such insertion sequences is discussed in detail in section 10.11. Even large insertion mutations can revert by further mutation, that is, by a deletion that removes the insertion. Other types of large-scale mutations seem to involve rearrangements brought about by mistakes and recombination. These include translocations in which a large section of chromosomal DNA is moved to a new location. In eukaryotes, often it is a different chromosome, and inversions, in which the orientation of a particular segment of DNA is reversed with respect to the surrounding DNA. The rates at which various kinds of mutations occur vary widely. Some types of mutations occur so rarely that they are almost impossible to detect, whereas others occur so frequently that they often present difficulties for an experimenter trying to maintain a genetically stable stock culture. Errors in DNA replication occur at a frequency of about 10 to the power of -7, -10 to the power of -11 base pair pairing during a single round of replication. A typical gene has about 1,000 base pairs, therefore the frequency of these errors in the gene would be 10 to the negative power of 4 and 10 to the negative power of 8 per generation. Thus, in normal, fully grown culture of organisms having approximately 10 to the power of 8 cells, there are probably a number of different mutants in each millimeter of culture. However, not all mutations happen at the same frequency. Transposition events may occur more frequently, at about 10 to the negative power of 4. On the other hand, the occurrence of a nonsense mutation is less frequent, 10 to the negative power of 6, -10 to the power negative power of 8, because only a few codons can mute into nonsense codons. There are also DNA sequences usually involved short repeats which are hotspots for mutations because the error frequency of DNA polymerase can be quite high at such sequences. Unless the mutant is selectable, the experimental detection of events of such rarity is difficult, and much of the skill of the microbiology geneticist is applied to increasing the efficiency of mutant detection. As we will see in the next section, it is possible to significantly increase the rate of mutation by the use of mutagenic treatments. In addition, we will also see that the mutation rate of a gene may change in certain situations. Mutations in RNA genomes. Whereas all cells have DNA as their genetic material, some viruses have RNA genomes. For example, these genomes can also mutate. Importantly, the mutation rate in RNA genomes is about 1,000-fold higher than in DNA genomes. At least some RNA polymerases have proofreading activities, like those of DNA polymerases. However, while there are several repair systems for DNA that can correct many changes before they become fixed as mutations, there seem to be no comparable RNA repair mechanisms. This very high rate of mutation in RNA viruses is not merely of academic interest. The RNA genome of viruses that cause disease can mutate very rapidly, presenting a constantly changing and evolving population virus. Chemical mutagens. An overview of some of the major chemical mutagens and their modes of action is given. Several classes of chemical mutagens exist. One variety of chemical mutagens are the base analogs, which resemble DNA purine and pyrimidine bases in structure yet show faulty pairing properties. When one of these base analogs is incorporated into the DNA, replication may occur normally most of the time, but occasionally copying errors occur, resulting in incorporation of the wrong base into the copied strand. During subsequent segregation of the strand, the mutation is revealed. Some other chemical mutagens react directly on DNA, causing chemical changes in one base or another, which results in faulty pairing or other changes. Alkylating agents such as nitroguanine or ample example are powerful mutagens and generally introduce mutations at a higher frequency than base analogs. Such chemicals differ in their action from the base analogs in that of the chemicals reacting on DNA are able to introduce direct changes even in non-replicating DNA, whereas the base analogs acts only after incorporating during replication. Both base analogs and alkylating agents tend to induce base pairing substitutions. One interesting group of chemicals, the acridines, are planar molecules that act as intercalating agents. These mutagens become inserted between two DNA base pairs, thereby pushing them apart. During replication, their abnormal conformation can lead to micro-insertions or micro-deletions, and acridine-treated DNA, thus acridines, can induce frameshift mutations. Ethidium bromide, which is often used to select DNA in electrophoresis, is also an intercalating agent that acts as a mutagen. Radiation. Several forms of radiation are highly mutagenic. We can divide mutagenic radiation into two main categories, ionizing and non-ionizing and electromagnetic. Although both kinds of radiation are used in microbiogenetics, non-ionizing radiations find the widest use and will be discussed. The purine and pyrimidine bases of the nucleic acids absorb ultraviolet UV radiation strongly, and the absorption maximum for DNA and RNA is at 260 nanometers. Proteins also absorb UV, but have a peak at 280 nanometers